Good morning, students. Yesterday we had the discussion with the herbal drugs, herbal formulations in our discussion, which we had some break by means of internet issues. But I told you to just go for my channel in YouTube, and also I gave the link, so you can go to the link and also you subscribe my channel and then you can take the new lectures whichever I am uploading. It will be helpful for you, and you can share with other your colleagues, whoever needs some doubts, and it will be helpful. Anyhow, we will go to the subject. Today we are going to discuss about the excipients which is used in herbal formulation and various formulations which we have come across. Yesterday we had a discussion about not herbal formulation, we had about the cosmetics, and in which even herbal cosmetics we did not go for various standardization and evaluation of herbal cosmetics, but it is also needed. But in practical sections we are coming across all the herbal cosmetics standardization, and as well as herbal cosmetics standardization, formulations standardization also we are coming across in the lab session, so which will be helpful for you in the theory also. So make yourself to have some discussion in all the detailed reference also by yourself in various internet source, or by even hard copy books from library also you can proceed. Please try to utilize the library also when you are getting time. Now you are busy, I know, but in this COVID situation, take care of yourself and also take care of your family, and wherever you go, with precautions you have to do and follow the protocol for saving ourselves and also doing some favor to our colleagues and to our lovable ones. Try to make everybody comfortable by means of sharing our love and affection, and to know about others' feeling and others' need. According to that we can move on. And now we are coming to this herbal excipients and herbal formulation. So anyhow, if at all, if we have any type of internet issue or any break like yesterday, you can, even I will try to upload this lecture also in the YouTube, and you can watch in the later. I will send a link for you. So today we are discussing about the herbal excipients and herbal formulations. So even here also in excipients we have to go through about various significant substances of natural origin excipients like colorants, sweeteners, binders, diluents, viscosity binders, disintegrants, and flavors and perfumes. And in herbal formulation, conventional herbal formulations like syrup, mixtures, tablets, and novel dosage forms like phytosomes. Some discussions, we are not going to deep, and it is necessary for you to apply your basic knowledge whatever you studied in your formulative pharmacy. It will be more helpful, and also when you are writing the exam it will be more you can impress the evaluator to get a very good score and good marks you can obtain. That's what my advice for the students. Now we are coming to the excipients, which we have seen. It is a big chapter when you studied in pharmaceutics, but so it will be easy for you to remember, and only thing is some of them are natural example. Apart from the synthetic sources we are just added, so we can know about the details of them. So excipient pharmaceutical aids are the substances which are inert and have little or more therapeutic value, or no therapeutic value, not more therapeutic value, no therapeutic value, because excipients are other than the active ingredients. That is what you might have studied in your pharmaceutics, but are essential in the manufacturing for various pharmaceutical dosage forms such as tablet, capsule and syrup. What is that? When you want to prepare a tablet, you need a binder, you need a disintegrating agent, you need for coating you need a coloring agent, you need a consistency, you need the shape, all these things. That is pharmaceutical applications. If you want to form a formula, if you want to make a formulation, you need some excipients to make them to. And for example, if you want to make a granules, you want to aggregate all the powder molecules to make a granules. If you want to make a suspension, you need the viscosity of the continuous phase should be increased to suspend the solid particles. If you want to have an emulsion, you want some surface acting agents, which is called, you can say that is surfactants, or you can say emulsifying agent, which will reduce the interfacial tension between the two immiscible liquids, and one will be dispersed in the other. So like that cream, and one would, yes, today some of the students who have come for the lab yesterday, I think A batch students, I told them what is the necessary, what is the difference between, ask them about what is some of the difference between cream and the lotion, and emulsion and suspension, and also what is the difference between one for example, apart from this semi-solid preparation, even for the liquid preparation also, what is the difference between elixir and solution, what is the difference between, and for example in the tablet also, what is the difference in the sustained release tablet and a normal tablet and a capsule, and also there is soft gelatin capsule, hot gelatin capsule. So if you see, there are various parameters which you want to talk about. For example, when you talk about soft gelatin and hot gelatin capsule, we will say the quality of the gelatin which is used, and also the product, the form of the capsule itself different, and also the drugs which are filled in the, or drugs which is supplied in that particular capsules are different. So according to the release, and according to the therapeutic use, and according to the formula, it is different. At the same time, when you see cream and ointment, it is about pharmaceutical application. Pharmaceutical, when you are doing how to prepare, for example, cream is an emulsion. If you are, for example, when you are saying about the ointment, one type of solid material or one powder can be incorporated in the ointment base. So, and cream can be compared with an emulsion, and also ointment can be compared with a suspension. A solid can be incorporated in the ointment medium. Like that, you can see, for example, liniment and lotion, by means of its usage it is differ. For example, liniment, you have to apply pressure when they are applying for any therapeutic use when you are using, but lotion you have to apply softly to the external area. Both of them are external preparation. Like that, there is lot of in their applications and their utilization, we are is having lot of differences, and usage of their therapeutic, usage of their excipients in the production of various formulation is needed for you to know about that. So that's what we have had a discussion yesterday. So these all things are general knowledge students should know, and so that they can write something very much attractive with knowledgeable questions. The answers can be prescribed in their answer book in the university examination. So excipients are mixed with the active ingredients to make up the volume to make up the volume. Sometimes if we want to increase the bulkiness, we are adding lactose, we are adding sometimes lactose, maltose in tablets and capsules to increase the bulkiness of the particular active ingredients. If the active ingredient is very small or very small quantity, it cannot be dispensed, so you need the increase the bulkiness. So you are using some powdery material which should not have any active therapeutic action. It should not have any therapeutic. That's why we are telling that it should not have any therapeutic action, because you are intended to have only one therapeutic action, which is the active constituents which you are adding in the formula. So that is why that is only the active constituents or active ingredients which you are intended to have that particular therapeutic action. They should have. Other excipients should not have any therapeutic action. So that is what the main idea. So you can see, and also sometimes we are using excipients to improve the stability and to mask the bitter taste and to improve the appearance and odor and other characteristic of the dosage forms. Binding agents, sometimes to bind you are using suspending agent, viscosity binders, disintegrating agent, surface acting agent, emulsifying agent, like that, etc. There are many pharmaceutical excipients which you are using, that is pharmaceutical aid. It is used for pharmaceutical aid. We used to say, what is the use of starch? It is a pharmaceutical aid, like that. What is the use of cellulose? It is pharmaceutical aid. What is the use of amaranth? It is pharmaceutical aid. What is the use? One you can use for many uses. So you can use for pharmaceutical aid. That is what we are using pharmaceutical aid. So advantages and significance of herbal excipients. That is biodegradable. Biodegradable means what? Biodegradable are the substance which can be easily taken back into the earth. It can decompose or it can be taken easily by the earth naturally without causing any harm to the environment. That is what. That is why we are telling, no, don't give plastic bags, don't give, and we give some that means some bags which can be easily disposed in the soil, and it will not make any issue for the environmental control. That is what biodegradable substance, especially the natural, when you are using excipients in any formula. That is why we are preferring natural components for any formulation instead of a synthetic compounds which you were using. Natural occurring substances show no adverse effect on the environment or other living beings, and they are easily biodegradable. And biocompatible or non-toxic is also very important and advantage of them. Most of the herbal excipients are carbohydrates in nature. They are compatible and non-toxic with most of the ingredient. So incompatibility with the main constituents will not be there. So you can go for the natural excipients for your production of any herbal formulation. That's what we are giving the advice. And economic, even it is economic value, you can see it has an high economic value. Herbal excipients are cheaper, and their cost of production is comparatively lesser. So it is very useful for economic value. Also economically it is useful for any type of producer. He can use for the natural source. And you see, safe and free from side effects. They are very safe and free from side effect. Naturally occurring excipients are safer and without any side effect. Easily available. Natural excipients produces in most of the countries, hence easily available. So it will be easily available for you to get them and access and you can further production. And disadvantage of herbal excipients, one is microbial contamination. During production, herbs are exposed to external environment, and hence there are changes in microbial contamination. That's why today also in the lab, I think in the section A students, we had a very good discussion about the microbial test which we did not perform for cream which we produced. Even I think almost all the batch students, all the 3 batch students have completed the production of cream, and we had mentioned that is microbial contamination have to be tested, and I told them you should know that what do you mean, what is the test we can carry out. It is a very simple, only 24 hours you have to keep, or you have to keep overnight, and next day you can come and observe whether there is any colonies have developed. We have to perform, so keep your cream safe and we will perform it as I told to them. To all the class students are listening now, so you can have this some idea which I am giving you. That means I remember I told about how to do this. That means there are 2 methods you might have studied in your microbiology. I asked them what is inoculation, needle. At the time also students were just in a blank stage, but it is a simple general knowledge, and it is very simple also. There is 2 methods. There are 2 methods of inoculation. One is you might have done by streak plate method. Another one is pour plate method. That what I told, because both of the method you need agar medium. So you are preparing an agar medium in the same manner. In a boiling tube you can prepare. In a boiling tube you are preparing a liquid when you are boiling with agar, and also what all the nutrients whichever needed you can add, and you are heating it, and when it is cooled down and you are pouring that into a petri dish, and it is solidified, it is cooled, and it is kept in refrigerator for 15 minutes, and then after solidifying you are just taking this plate to the inside the aseptic chamber or in the front of laminar flow. You are taking an organism from the slant culture which is already kept in the aseptic manner. You will be inoculate. Instead of that, you will inoculate the cream which you have produced by means of taking a small bit of cream by means of your inoculating needle, and make a streak over the plate. Just you are making a streak. The plate will be carried like this, and you are making a streak over a line by means of the inoculating needle by touching your cream, and so that, and close down in an aseptic manner with then 2 lid. You can keep the cover lid of the petri dish and keep in the incubator for 24 hours in 37 degrees centigrade plus or minus 0.5 adjustment, and keep for 24 hours, and then you can come and observe in the petri dish, and if you see there is in the streak which you made, if there is any growth of microorganism, then you can say that is your cream is having contaminated by means of the microbial growth. That is why there is even the microbial study will give you just a pass or test. And the same thing, what do you mean by pour plate method? Same, it's also the same. Only thing is, what is the difference? Boiling tube which you are doing, boiling it with an agar medium and nutrient agar medium which you are boiling in a hot condition, and you are bringing it in a liquid form. You are not allowing it to get solidified, or you are not pouring it directly into the petri dish. You are inoculating your cream inside this liquid form only. You are taking this boiling tube inside that aseptic chamber or in the laminar flow. Aseptically, you are just by means of inoculating needle, you are inoculating that cream inside this liquid agar medium, and after this liquid inoculated, this liquid agar medium in the aseptic manner, it is transferred to the petri dish in a liquid form itself. After inoculation, this is after inoculation you are transferring to the petri dish. There medium is transferred to the petri dish, solidified, and then making streak. That is the only difference. And after pouring this will be get solidified by means of keeping inside the fridge, and then you are in a low temperature, not very low temperature. Just you should not get freezer, you should not keep, or you can even keep outside for solidification. After solidification, you keep it the lid and just keep in the inoculating chamber or in the incubator for 24 hours in 37.5 degrees centigrade plus or minus 0.5 degrees centigrade. You can maintain for 24 hours, and you can see if there is any colonies uniformly it is grown in the agar medium, then you can say that our cream is having some microbial organism, microbial contamination. If there is nothing is there, then you can say that our cream is microbial free. That is what we have discussed today in the class. So this will be useful for the student. You can perform this, and also you can write Both the methods can be followed for finding out whether our product is having microbial contamination or not. This is the test which we are telling. Always it is having some microbial contamination may occur because when you are using certain herbal formulations, when you are using. And biochemical variations, you can see variation of quality of the product may occur due to variation in environmental factors, because they are growing in the soil in different area that may be affected, and also uncontrollable rate of hydration. This occurs due to biochemical variation in difference in the quality of the material from one batch to other. And heavy metal contamination is also sometimes possible. Herbs are always associated with the risk of heavy metal contamination. Note, microbiological contamination occurs due to improper processing as well as the presence of excessive amount of moisture in the raw material. It may occur for microbial contamination. That is what we want to see, microbial contamination, very important for any formulation which you have made as a herbal formulation. And you can see the classification of herbal excipients. Herbal excipients can be broadly classified into following categories based on their function. Herbal excipients, example, colorants, sweeteners, binding agents, diluents, viscosity binders, disintegrating agents, ointment bases, emulsifying agent, flavoring agents. These all are some of the excipients which I have listed out. And you can see colorants, example, I am giving only example, but you should know about some more details because you have studied in your formative. That is why we are not going to give more details. But colorants, what is the advantage? What are the things which will give for pharmaceutical aid? These things you have to explain if they ask sometimes an essay question. So here colorants example, I am giving only the example like henna, chlorophyll of the leaf, and also that is caramel, amaranth, indigo. These all are the examples. Sweetening agent example, glycyrrhiza glabra, glycyrrhiza, honey, and stevia. Stevia is also in Malayalam, it is also called stevia also they used to say, sometimes Madura, and Madura in Tamil it is called Madura Kira, or in Madura Chira can also be told in Malayalam. So it is also having some sweetening agent which is commonly available. And the binding agent, acacia, gelatin, and tragagan, starch. And diluents, lactose, starch, mannitol, sucrose. That is diluents which you want to increase the bulkiness of the active principle. We used to use lactose, starch, mannitol, sucrose. These all are naturally available components. Viscosity binders, pectin, tragacanth, cellulose, guar gum, gelatin. So next one is disintegrating agent. Disintegrating agent is very important for any type of unit dosage form like capsule, tablet, especially because it should disintegrate into smaller particles, then only the dissolution will take place and absorption, and or the other next the absorption, then comes the for various distribution will take place. This initial stage is disintegration is very important. When you studied about the pharmacokinetics, especially you will say first process is disintegration, then only the dissolution will take place, distribution will take place, and absorption, and then distribution will take place. So that is a disintegration of the very important, like starch, ispagul husk, and the carboxymethyl cellulose are also can be used for this purpose. And ointment bases, as I told, bases lanolin, beeswax, they are the basic components which is mainly depending on the natural sources. And also emulsifying agent, acacia, agar, guar gum, methyl cellulose, these all acts as an emulsifying agent. They are the surface acting agent. They may reduce the surface interfacial tension between the two immiscible liquids if they are interested to make as emulsion. And flavoring agent, cardamom, vanilla, lemon oil, orange oil, and perfumes like rose, lavender, sandalwood, these all can be used as a component of excipients. And a study of the herbal use as excipients, and first example henna, the source it contains of dried leaf of Lavasonia inermis, belongs to the family Lytaceae, and chemical constituents henna contains lavasone as its chief constituents. Other constituents include phenols, coumarins, flavonoids and tannins. Use of henna is coloring agent, and there are many other uses in cosmetics, but still you can see as an excipients it can be used as a coloring agent. We are using for hair dye also. There is very commonly there is available henna is very commonly used. Amaranth we are using as a coloring agent. It is also a natural source. It contains the flowers of amaranthus, Coriancus. So you can see the other species of amaranthus belonging to the family Amaranthaceae, and the chemical constituents it contains polyphenols, vitamins and flavonoids, and used as a dyeing agent in various sectors. And also liquorize, we know very commonly liquorize, already we come across in liquorize, we studied about liquorize in our other classes also. We have come across in previous in 5th sem itself we have studied liquorice, that is Adi Maduram in Malayalam, and also it is used in various components of in Ayurveda and Siddha preparations, and it contains of dried root and stolon of Glyceris glabra belonging to the family Leguminosae, and chemical constituents it contains glyceric acid or glycerinic acid and licuritin, isolicuritin, and used as a sweetening agent. It is used as a sweetening agent. You can see the picture which I have shown there. It will be helpful for you to remember about liquorice. And as I told, this is also available in Kerala, and it is called Madura Chedi, or you can say Madura Kirai. Sometimes it is called in Tamil. And whenever it is in Trivandrum side, we used to say Chirai, that is Madura Chirai, Madura Kirai, or Madura Chedi also can be called as. So it is called stevia, and it consists of plant stevia, that is Ribodinia, and belonging to the family Compositae. And chemical constituents it contains stevioside, ribosidioside, and stevol, and uses it is a sweetening agent is used. And about now acacia, very commonly you might have used many times acacia in pharmaceutical preparations, and when you are doing in pharmaceutics, and also even in the formulative subject you might have done. Source, it is a dried gummy exudation obtained from the stem and the branches of Acacia arabica belonging to the family Leguminosae. Chemical constituents, it contains sugars like arabinose, galactose, ramenose, and glycerinic acid. Uses, acacia is used as binding agent, suspending agent, emulsifying agent, and viscosity binder. It can be used as a viscosity binder, viscosity builder. Emulsifying agent, and it can increase the viscosity, and for suspensions it can be used. Emulsifying it can be done by means of reducing the interfacial tension between the two immiscible liquids so that it will act as an emulsifying agent. Suspending agent by means of increasing its viscosity so that suspend the solid material in a liquid can it's all possible. And also binding agent, you can see it can be used as a binding agent also in various solid dosage forms it can be used. And you can see in gelatin is having more use, even soft gelatin capsules, hard gelatin capsule, they all depend on the gelatin. Gelatin production itself a very big production in our pharmaceutical field is a bulk production. It is also a good technology which we have to study about how gelatin is produced, and the source it's a production, it's a protein, not production, sorry, it's a protein obtained by partial hydrolysis of animal connective tissue like bone, skin, tendons, and ligaments. And chemical constituents, gelatin chemically contain amino acids like glycine, alanine, glutamic acid, proline, arginine, aspartic acid, leucine, and isoleucine. It is used in the binding agent and thickening agent, emulsifying agent, and in the manufacturing of capsules. The picture which I have shown is just to make it the case. This gelatin is available in the form of powder, it is available in the form of square cube gels, and it is available in the form of capsule, soft gelatin capsule. It is available in the form of hard gelatin capsules, empty capsules, which are also available. And Tragaganth, if you see Tragaganth, shows Tragaganth is also a very important plant. You can see this plant is also available in it's available in our place. 1 or 2 trees will be available. It has fruits will be like this. You can see, you may not be noticed, but it is available most of the place in the ground area, in our schools. Even in our school I have seen this Tragaganth is available. It has some gummy exudate which is obtained. It is an unorganized drug which can be produced from that particular trunk of that particular plant in the various area. We can make, like acacia, how they are collecting the exudate of the gum, which same type you can see the crystals of the Tragaganth which is shown in the picture. So it is a dried gummy exudation obtained from the stem and the branch of Astragalus gummifer, family Leguminosae, belonging to the family Leguminosae. Chemical constituents, it contains gums, and also it has a constituents especially Tragaganthine and basorin. And uses, it is used as a binding agent, and also it is increases the viscosity builder in various formulation like suspension. It can be used as a viscosity builder. Lactose is you can see it is lactose is one of the component which is very commonly used for diluent and to increase the bulkiness of various formulation like tablet. So what is the use, what lactose, even it is having more uses in it's a carbohydrate, it has more nutrient effect and also ingredient, also it is not having any therapeutic effect which can be easily added for any potent drug, which is if it is very low quantity we can increase the bulk by means of lactose. So it is a natural disaccharide obtained from milk, and chemical constituent, it contains sugars, galactose and glucose. It is used as a diluent in the tablet and the capsule. It is very commonly used. Mannitol is also one type of sweetening agent. It is a saccharin exudation from the stems of the Fraxinus ornus, belonging to the family Oleaceae. It is a white, crystalline, odorless, non-hygroscopic sweet powder. It is freely soluble in water and insoluble in alcohol. Uses, it is used as a sweetening agent and as a diluent in various preparations. And you see guargum, it is also in Malayalam we can say that it is called, sometimes it is called they used to keep some not curry, they used to they used to make, sometimes this is very commonly used, and it is used for diabetics and also even liver problems. It can reduce, even it can stimulate the pancreas. It has more medicinal value, and so we are using it what we can say, some vegetarian foods they used to make this as a tuer, and it can be made, and it is very commonly used, and not very bitter, slightly how the preparation, when you prepare properly the bitterness may not be seen, but it is a good for health. And it is a ground endosperm of the seed or seeds of, we are using guargum is taken from the seeds, but we are taking the fruit before fully ripe in a raw stage, in a bud stage. It will be very tasty, and it is called Cymopis tetragonolobus or gonolobus sometimes, or sometimes in gonolobo or gonoloba is also in many places they are using. It is called Cymopis tetragonoloba or gonolobus. Usually in certain books gonoloba they are telling. It is belonging to the family Leguminosae, and the chemical constituents, it contains 85% of guaran, galactose, maltose. It is used as a binding agent, disintegrating agent. It can swell and it can break down all the tablet. Even if you make a herbal tablet, it can use as a disintegrating agent, suspending agent and emulsifying agent. It has an effecting of suspending agent, emulsifying agent. So guargum, it has a more pharmaceutical aid of its use. You can see that. I hope when you see the picture of the green color which is shown in this here, it will be helpful for you to remember what you have done in the in your home. You have come across these type of vegetable you might have come across in your home. And starch, you know very well, we are using starch for pharmaceutical aid in various formulation. We are using starch. Source, it consists of polysaccharides granules obtained from the grains of maize starch, rice starch, wheat starch, potato starch, and even tobacco starch. Like for example, when you see our very common kappa is having a very good, that means we can say it is called Keranga, that means you can see that is in certain places one name. Even you see in the southern part of the Kerala, we can say it is called Marichini Keranga. So that is also one type of, it is having more rich potato starch is also available. And you can see chemical constituents, it contains water soluble amylase and water insoluble amylopectin starch structure, which swells and responsible for gelatinizing property of the starch. So uses, it is a binding agent, disintegrating agent, and diluent. It is also used as a diluent. It is also heavy pharmaceutical aid use in all the formulation. It is useful, the starch. And sandalwood oil, you see sandalwood oil, sandal is also for cosmetic, we are using various purposes. Sandalwood oil, as shown, it is also costly oil. Source, it is an oil which is obtained from the steam distillation of hot wood of Santalum album, obtained or belonging to the family Santalaceae. And chemical constituents, it consists of sesquiterpene alcohol, namely alpha and beta santalol use. It is used as in perfumery industry. And rose oil, what is the use of rose oil? Source, it is an volatile oil distilled from fresh flowers of Rosa gallica and other rose species belonging to family Rosaceae. Chemical constituents, rose oil contains lanolin, nerol and citronellol and geraniol. It is contained and used in the perfume of flavoring agent. You can see rose oil very commonly we are also using in our lab. And cardamom is very common in our area. In our district, in Idukki district, we have cardamom cultivation. Cardamom can grow very easily for this climate. And the source, it is a volatile oil distilled from the seeds of the Elateria cardamomum belonging to the family Gingiberaceae. Chemical constituents, it consists of cineol, alpha, terpenyl acetate, and terpeneol, boreneol, and sabinine, and it is used in as a flavoring agent. So this is the cardamom. We can see that fruits which is coming in the below the shoot system, which we can find out. This is the which is very commonly known product. And orange oil, source, it is a volatile oil obtained by expression of the fresh peel of the ripe fruits of citrus Lemonia and belonging to the family Rutaceae. Chemical constituents for orange oil contains limonene, citronellol and citronellol, and it is used as a flavoring agent. And now we are coming to the herbal formulation. Even in the formulation also, I am giving only about some definitions and some small things, but you have to use your knowledge whichever you studied in your formulative and pharmaceutics. You can elaborate the formulations, what all the evaluation techniques, what all the processing in the quality control and quality assurance techniques, whatever you studied in your pharmaceutical subject, you have to incorporate here also. And now we are just going to discuss about some of the few things in herbal formulation. Herbs have been used in a wide variety of dosage forms. Large number of herbal formulation containing the plants of their extracts with proven medicinal activity are being used. With an advantage of pharmaceutical technology, modern dosage forms have evolved. The herbal formulation dosage forms can be broadly classified into three categories. That is, traditional dosage forms, which we know that there are many traditional dosage forms like tablet, pills, and also even herbal formulation like Arista, Swa, Legia, pills. Before tablet it is pills, that is all traditional, and the syrup, liquids, and also creams also, and ointments like that. Modern herbal dosage forms, there are many modern herbal dosage forms like patches, transdermal patches, and there are many other things are there. Even my very application by means of in the skin, scraping the skin and also applying as an, and also implant. There are many formulation, modern herbal formulation dosage forms are there. And even many novel drug delivery system, that is NDDS, novel drug dosage form, is a new technology which you might have studied in pharmaceutics, and formulating you have a subject itself, so it will be easy for you to write these definitions. That means very micro, that means a micro encapsulation is also comes under this section, burst micro encapsulation. Then that encapsulation, how you are making by chemical micro encapsulation, physical micro encapsulation, these all you might have studied, chemical and bio and physical encapsulation of micro encapsulation you might have studied in your pharmaceutics or in the formulative pharmacy. So you know the technology which is very commonly used even for traditionally for sustained release of the drug, which controlled by means of various matrix and polymers, and that is lysosomes, which you have been used for various purposes in your pharmaceutics you might have studied. So that's what we are going to discuss, some of the things in this area. And note, herbal formulation may contain a single herb or combination of different herbs are also there. So one traditional dosage form, these are derived from various traditional forms of medicine like Ayurveda, Unani, homeopathy, etc. For example, pills, powders, semi-fluid extracts, pellets, tinctures, etc. And these definitions you may be knowing, or you have to know about that, what are they. Modern herbal drug forms, now these forms develop a modern technology process. Modern herbal formulation offer small dosage form size and also very user friendly, convenient to have, good absorption characters and distribution characters, for even for the action also will be improved, like tablet, capsule, syrup, solution, suppositories, even injections are also available nowadays. And also novel drug delivery system, NDDS, novel dosage forms. So with an advancement in different scientific technique of preparing formulation, novel dosage forms are being developed to overcome the limitations of conventional dosage forms such as tablet, syrups, solution, etc. Many novel dosage forms have been developed successfully, which have offered better acceptance by the health system. A few novel dosage forms available in the market are transdermal patches, implants, nasal systems, microcapsules, microspheres, lysosomes, phytosomes, etc. So these all are some of the novel drug forms. Many novel drug forms, even patches also included. I was telling that it is modern, and now it is also it is advanced release drug, which is novel drug forms. It nano particles also involved in this process. So by means of a simple classification, herbal formulation can be divided into syrups, tablets, ointments, creams, mixtures, and novel dosage forms like phytosomes, liposomes, nanoparticles, microspheres, which can be incorporated in various formulation like patches and transdermal preparations can also be included in this novel dosage forms by using this liposomes, nanoparticles, microspheres. And you can see herbal syrups. I have given only the examples. You should know how to prepare and standardize these all things. We have to utilize. You have to, whatever you have studied, whatever you are making in the practical session is also important for your theory, and to improve your more references and do the things very successful when you are writing the university exam. These are preparation formulated by incorporating sugar solution with plant extracts such as infusion, decoction, juices, fermented products or simple solution. Honey or unrefined sugar is used to prepare syrups as they act as good preservatives. Herbal syrups are made with equal proportions of herbal extracts with honey or sugar solution of known concentration. Various flavors like orange, raspberry, mentha can also be added to mask the bitter taste as well as improve the palatability of the formulation can also be improved. So some example, like I have shown some oils, some cough syrups, and also you can see there are syrups, that is thorax. These all are herbal syrups are also available in the market. Tablets, these are solid dosage forms of powdered herbal extracts or their constituents prepared by molding or compression. You know that molding by means of using some mold, solidification can be done like previous method, but compression, you have studied more compression methods, single punch method, rotary press method. So there is lot of compression methods are there. So for compressing tablets you need made to granules, and then you have many methods you might have studied. That is wet granulation method, dry granulation method, direct compaction method. So all methods can be applied. Direct compression method means all material cannot be compressed directly. You have the ingredients without any type of cannot be used moisture cannot be used for the certain components, so that you can go for direct compaction method. And dry granules are pellets which can be made and then crushed into granules without adding any type of water or anything. It is also possible direct, that is dry granulation method, and compression can be done. Wet granulation method, adding binders in the form of solution or a paste, starch paste, and then drying by fluidized bed dryer, making granules, and then you are taking to the compression area and compressing tablets. And then you can go for coating, film coating, sugar coating. There are many various coating also you might have studied that can be also done here. And parameters, you should know that tablets you have many parameters. What are the things for evaluation you can do? You have to write in the in process check what you are doing, like hardness, shape of the dimension tablets, and also you have to see the disintegration test, dissolution test, and also various condensed test can also be carried out for, even if it is an coated, then you have to see the coating, what is its mottling, what is the tensile strength of the coating material can also be seen, observed, and can be checked before product have been released from the quality control for the market. So in addition to the active ingredients, these contain diluents like binding agents, and they have many pharmaceutical substances which provide strength to withstand normal handle within to transport and storage, and coloring agent to improve the appearance, sweetening agent and flavoring agent to mask the bitter taste, and disintegrating agent to facilitate the disintegration and breakdown for absorption in the gastrointestinal tract for the various therapeutic action. I just stopped by means of only one slide, but it has many parameters which have to be incorporated by the student, and also by means of reference you have to add, and by means of your knowledge what you have by means of the pharmaceutics you have to add in here also, so that it will be enlarged and also it will be useful for you to answer the final examination paper. Very intelligently you can do it. by means of your basic knowledge which you got from your pharmaceutics. And now we are going to the ointments. Herbal ointment, these are semi-solid dosage meant for the external application to the skin mucous membrane. Ointment perform softening, emollient and protective action. Waxes like beeswax, paraffin wax are used in the base or carrier. Various active herbal ingredients in the form of powders, dried extracts can be incorporated in the bases which provide therapeutic benefits. So that is an example for herbal ointments. And creams, which we have done in your practical, we have done the cream. So you see, these are viscous semi-solid preparation which may be oil in water type aqueous creams or water in oil type oily cream. We have also done same cream by means of aloe vera, which can also be done, like emulsions can be done with a careful of taking in the mortar pestle. The emulsifying agent and the oil phase will be made into, first if you are using oil in water emulsion, add the oil phase and the emulsifying agent, triturate in the same direction, and also make, after getting the clicking sound of the preparation, add the aqueous phase slowly and make its consistency to get a cream properly. You can make very intelligently how your skill work can give you very good product of cream. Herbal mixtures, these are the preparation containing combination of 2 or more herbal ingredients which are formulated into various formulations like tablet, capsules, ointments, creams, etc. These mixtures are combination of herbal ingredients, may have more than one active and can be used for the treatment multiple ailments, or they may produce additive or synergetic effect of a single disease. So, if there is many combined form, you can see even Arista in Ayurveda, they used to add in various ailments, and also it has, sometimes you have to be careful, you have to see that what is the multiple ailment it can be used at the time. Sometimes additive effect and synergetic effect may occur. So, what do you mean by additive effect? 1 plus 1 is equal to 2. Synergetic effect is not like that. You might have studied about synergetic effect explanation in the pharmacology. You might have studied synergetic effect between, especially I do remember when you are adding alcohol with phenobarbital, they will give synergetic effect of sedative. The person will go for even up also possible sleep, a long sleep will be there because of its synergetic effect. Additive effect 1 plus 1 is equal to 2, but a synergetic effect is 1 plus 1 is not 2. It will become 3, 4, 5, 6 and 10, even 12. It will be increasing multiples. So that is called synergetic effect. And next, novel drug delivery system, NDDS, novel drug delivery form, that is dosage forms. A, that is phytosomes. Phytosomes, you see the picture? See the picture, inside there is a ball which is showing that active, that is herbal formulation, is surrounded by polylipids, curcumin or that is phospholipids, which can be surrounded in a small, it is a very small micromolecules which we are showing, so that it release can be controlled. That is what this concept of phytosomes is another breakthrough for the development of herbal drug technology. It is a very important herbal drug technology about phytosomes. Phytosomes contain active herbal ingredients surrounded by, see the picture, surrounded and bounded by phospholipids. They are produced by bounding phosphatidyl choline with an ingredient, thereby producing a complex. So the phospholipid structure has a water soluble choline head. You can see that head and a tail, whole head, and a fat soluble body tail, that is phosphatidylate portion. Water soluble head and fat soluble body and the tail. Fat soluble body and the tail, that is phosphatidylate portion, which is coming to the herbal portion, and the water soluble portion is in outside. Understand? Can you follow? That is what we can see. So solubilize and release. So control of the herbal drug will take place. Normal dosage form usually contain pure isolated compound herbal nanoparticles or colloidal system with particle size of about 1 to 1000 nanometer. Very small. And the choline head, water soluble, binds with an active compounds herbal ingredients, while the phosphatidylate portion, fat soluble, that is in the fat soluble, which is in the tail and the body portion, bounded material, that is active ingredients as a result of microsphere or vesicles are produced, whereby the valuable compounds of the herbs are protected. Valuable compounds of the herbs are protected. So the tail and the body, which is having the fat soluble components, which will protect the first layer, and the outer layer will be water soluble layer will be there, phospholipids, which can also be produced by means the choline can also protect the herbal drugs, which are very constituents which is present inside the particular small sphere. That is, phytosomes enhances the absorption and improve the bioavailability of the herbal ingredients by enhancing their delivery into the tissue, thereby lowering its dose and reducing its side effect. Dose is reduced means automatically the side effect is also reduced, but there is usually they have less side effect. There is no side effect at all, you can say. That is, if you are using in smaller dosage which is required, the dosage plays a major role for every, not only herbal drugs, every medicine's dosage plays a major role. They also protect the active herbal ingredients from destruction by the digestive juice and gut bacteria. Several studies have shown that the body uses phytosome molecules more effectively than that of non-phytosome molecules. So phytosome molecules is better for any therapeutic use. So nowadays normal drug production, this is a new technology which is very commonly used for herbal drugs, for phytosome molecule preparation. Preparation of phytosomes are generally prepared by adding accurate amount of phospholipids, that is soya, lecithin and herbal extract in an appropriate solvent. Soya lecithin contain a main constituents of phosphatidylcholine, which is having a double function. Phosphatidyl part is lipophilic in nature and choline part is hydrophilic in nature. So the choline part is attached by the hydrophilic main active ingredients, and whereas the phosphatidyl part of lipid soluble component attached to the choline bound complex. So this results the formation of lipid complex with better stability and bioavailability of the components. So you can see that phytosomes can be prepared by one of the following methods. Before that, you should know very clearly about what is the water soluble part and what is the lipophilic part of an phytosomes. By the picture which I have shown before, you have to go through that so that you will not find any difficult to write this question very clearly. With your pure understanding, it is possible for you to write more bigger answers with clear detailed explanation can be given. I am confident if you see that definition about what is that hydrophilic and lipophilic solid part of the phytosomes, especially the molecules, the herbal constituents or the herbal active principle inside is surrounded by the phospholipids, especially the choline part. They have a head and tail. That means the tail and body is aqueous portion, and the head that is hydrophilic portion, and these portions are completely supplied by this component when you are preparing what you are using, that is soya lecithin will give you it will give you the complex, and so that you can prepare the phytosome very easily. So there are three methods. One is anti-solvent precipitation technique. The specific amount of drug and the soya lecithin was taken in a 100 ml round bottom flask and reflects with 20 ml of dichloromethane at a temperature not exceeding 60 degrees centigrade for 2 hours. Very easy, you can remember, very simple. Whatever, you are not adding more components, you are adding very little components. Please read once and make a hints, and you can write all the three, and very clearly you can write, and you can get the full mark from the evaluator. The mixture is concentrated to 5 to 10 ml. Then hexane 20 ml was added carefully or by continuous stirring to get a precipitate, which are filtered and collected and stored in a vacuum desiccator overnight, and the dried precipitate was crushed in a mortar and passed through a sieve, and a standard IP standard sieve 100 mesh, and the powdered complex was placed in an amber colored bottle and stored in a room temperature. That is the first method, and in this method the picture will give you very detailed what I explained in the previously. You see that? And here you are taking the drug soya lecithin 20 ml of dichloromethane, heating for only 60 degrees centigrade for 2 hours, and then what you are doing, in concentrating to 5 to 10 times, and plus you are adding 20 ml of hexane, continuously stirring, you will get the precipitate. Filter it and keep it in vacuum desiccator overnight and dry the precipitate. Bring it to the sieve for 100 mesh according to the IP standard, and then amber colored bottle complex have been stored and in a well closed container. Finished. This is what very simple production. And also second method is also same thing. There is no much difference. Only thing is you have to remember tetrahydrofuran we are adding. All the other things are same. The specific amount of drug, you are taking the drug which you are interested to prepare this phytosome, you are taking the drug, and soya lecithin is the same component and dissolved in 30 ml of tetrahydrofuran in a rotary round bottom flask, and followed by stirring for 3 hours at a temperature not exceeding 40 degrees centigrade. Thin film of sample was obtained, to which N-hexane you are adding, N-hexane and was added continuously stirring using a magnetic stirrer. You know that magnetic stirrer, and the precipitate obtained was collected and placed in an amber colored glass bottle and stored at room temperature. Same, only thing is, you see, magnetic stirrer you are using and stirring it. Drug soya lecithin was dissolved in 30 ml of tetrafluorofuran and then stirred for 3 hours. Temperature should not be more than 40 degrees, and thin film of sample and hexane was added, continuously stirring, and then the precipitate is stored in an amber colored bottle in a room temperature as shown in the picture. And the third one, solvent evaporation method. Here also you see soya lecithin. Only thing is, you are just having a small difference. There is nothing. What is that? You are just specified amount of drug, and soya lecithin is taken in 100 ml of round bottom flask and reflects for 20 ml of acetone. Here acetone you are adding solvent evaporation method. 20 ml, it is not M1, ml, that is typing error. ml, 20 ml of acetone at a temperature of 5 to 60 degrees centigrade for 2 hours. The mixture is concentrated to 5 to 10 ml and obtained precipitate and filtered and collected. Directly acetone, that is solvent evaporation, precipitate you got, and the dried precipitate complex was placed in an amber colored bottle. No need to add hexane, no need to have anything, precipitation is formed, only solvent evaporation method in a room temperature you can collect. You see the picture I have shown, the same thing. Drug and soya lecithin was taken in a 100 ml round bottom flask, reflex, heat it 60 degrees centigrade for 50 to 2 hours, and add 20 ml of acetone and concentrate 5 to 10 and obtain a precipitate immediately and filter and collect the precipitate. That precipitate will be stored in the bottle and a room temperature amber colored bottle. So very simple, all the three. And now the second one is phytosomes. It is first we have 3 steps, and you can see liposomes. You might have studied in your, even in your 7th sem you have the subject, I think so, you might have studied the new technology obtained. So this is simply a definition for you, very easy. These are prepared by incorporating the active ingredient inside the microscopic double layer as shown in the picture. That means hydrophilic head, hydrophilic tail and aqueous interior, and you are having that component inside, that unilaminar liposomes, and this layer member which are made up of phospholipids known as phospholipid vesicles. The vesicles are suspended in an aqueous solvent uniformly. Drug is incorporated in the liposomes in the picture as I shown, can be delivered in the desired site, desired concentration for a long, for example, and patients like arthritis patient, cancer patients, they need drugs for hours together to be released, because their pain is not to be subsided by means of immediately, it will come again and again. So they need drug, minimum effective concentration should be maintained in the blood for a long time. So in that case, this type of drug for a sustained release and for a prolonged release, and the release can be controlled slowly, that the drug is incorporated in the lysosomes, can be delivered to desired site of desired concentration. This novel drug delivery system is especially targeted by liposomal delivery of drug in cancer therapy, arthritis and hemophilia and disease of immune system. These all drugs are very commonly useful for various systems even in the cancer treatment. And nanoparticles, very small minute particles, nano, it is nanoparticles, 2 nano means very small. So these are colloidal particles of sub-micron size, which acts as carrier of drug and carrier of drug. It is carrier of drug molecule. These are used to target various sites in the treatment of cancer disease and in the reticuloendothelial system and enzyme replacement therapy in liver. So this is also even replacement therapy in liver. This can be nanoparticles, which is even herbal also can be incorporated. Microspheres, it is an advanced, it is the same thing which you have studied in microencapsulation, which is very commonly pesticides also they are preparing by this method for releasing of the drug in slowly releasing of the chemicals, organophosphorus chemicals, which is used for that is as a pesticides, they used to use these chemicals. So microspheres, these are small solid particulates, carriers containing a dispersed drug particles either in solution or crystalline form. In a chemical microencapsulation, you might have studied how they are preparing droplets with covered by sometimes matrix, physical method also there, chemical method also there. So that can also be utilized for these microspheres are used to carry for drugs and therapeutic agent, especially in cancer treatment and hormonal disorder. Like that, there will be a shell with the polymer 1 and the core polymer 2 will be dispersed of the drug. The drug will be released by the number 2 polymer which is present inside, and then it will be coming out. The coat will be removed slowly. That is called, you can see it's a micro, very microspheres also preparation can be done. So in this all we have discussed about most of the herbal formulation, and only the formulations definitions I have given. Apart from that, you have to use your theoretical and the basic knowledge whatever you have come across in your formulative subject and pharmaceutics and also formulative pharmacy subject. You have to incorporate and you have to increase your references and make your study very feasible for writing your exam. And thank you very much for being with me. Anyhow, I hope the class will be useful, and this will be available in the YouTube in my channel. You can subscribe and you can download, or you can even live, you can listen and even you can download and you can keep anytime. Whenever you get time, please listen, and if it is useful, thank you, have a great day.